Hi, my name's Lauren Lynch and I'm going to present my PowerPoint presentation on human immunity. So, what is an infection? An infection is caused when a pathogen enters the body. It can result in a fever and other health problems depending on where it occurs in the body and on the affecting pathogen. The human immune system defends the body from infection or disease caused by pathogens. If we do become infected, then the immune system can destroy the pathogen and in most cases protect a person from illness if we are infected by the same pathogen again. Pathogens can be bacteria, viruses or fungi. They can all cause illnesses and disease. Let's take a look at each one. So bacteria such as food poisoning, viruses such as influenza and COVID-19, fungi such as thrush and athlete's foot. Pathogens can be transmitted from one person to another. Pregnant women can spread pathogens to an unborn baby via the placenta or by breast milk. Pathogens can also be spread by eating contaminated food and drinking contaminated water and through animals. So how does the body defend itself? The body has specific and non-specific lines of defence. I will look at non-specific defences in which there are two responses. The first line of defence are chemical and physical barriers that do not distinguish between specific pathogens. They act in the same way whatever the type of pathogen it is. So let's take a look at these barriers and how they can prevent infection. So we have skin, covers all of the body and acts as a physical barrier that pathogens can't cross. They can, however, enter through cuts in the skin. It's hard to penetrate waterproof barriers. Glands in the skin secrete oil, which kill microbes. Then we have eyelashes. They stop pathogens by trapping them and wafting them away. Cilia, small hairs. Cilia are small hairs that are found in the nose and the throat. They trap pathogens that may try to enter the body. They are then coughed or sneezed up or blown out of the nose. Genital and urinary tracts. Harmless bacteria in the genital tract together with mucus prevent pathogens from entering the body. Tears and sweat. Tears contain enzymes and salt, also found in sweat, which can destroy pathogens. We have mucus, which is found in the nose and throat, and this is a thick liquid-like substance that traps pathogens inside it. Stomach acid, so the hydraulic acid with a pH level of 2. Microbes are unable to survive in such acidic conditions and enzymes produced in the stomach can kill the pathogens. Urine washes microbes out of the urinary tract. So the second line of non-specific defence mechanism involves responses within the body if the pathogen can enter it. The inflammatory response is the body's first response to infection. The blood supply to the area affected will increase so that cells can destroy the pathogens, can reach out to the area quickly. There is also an increase in temperature and the area may become red and swollen as the pathogens are destroyed, pus is formed and then they break down. So let's take a look at a fever, for example. A fever may develop in the rest of the body as some pathogens cannot live above certain temperatures and are killed. These cells found in the blood are generally known as white blood cells. There are several different types. We have neutrophils, and these cells can move by the process of chemotaxis through the blood vessels and are usually the first cells to arrive at a site of inflammation or infection. They engulf the invading organism via phagocytosis. They engulf it and then using the enzymes to destroy the pathogen. Cytokines release chemicals which are used to attract other immune cells to the area and other small proteins called complement proteins which can destroy the pathogens. Cytokines can cause a cascade reaction. They stimulate the production and release more cytokines. There are different types of cytokines which target different immune cells within the blood. So moving on to monocytes and macrophages. Now these are the cells found in the blood. Once monocytes arrive in an infection site, they undergo changes and then they become macrophages. These engulf pathogens via phagocytosis and once ingested, enzymes inside destroy the pathogen or foreign particle. As you can see on the slide here, so monocytes arrive at the infection, they undergo the changes, then they become the macrophages. Pathogens are engulfed via phagocytosis, enzymes destroy the pathogen, some macrophages can destroy dead or damaged cells, and macrophages are found all over the body. So let's take a look at antigens and pathogens. If pathogens such as bacteria and viruses manage to invade the non-specific immune responses, they will come up against our third line of defence, the specific immune system response. This is the body's response to specific pathogens. Pathogens have substances on their outer surfaces which are specific to them. They're called antigens and they can help the pathogen infect the body by entering the cells. The body can produce substances called antibodies which then join to each specific antigen on the pathogen. This leads to acquired immunity. It allows our immune system to react quickly to future infections to destroy the pathogens as it remembers the pathogen and produces the relevant specific antibody to destroy them. It's also known as natural acquired immunity. So let's take a look at artificial acquired immunity. We have eosinophils. These cells of our immune system which fight infection are found in the blood and are genuinely called white blood cells, but there are 5 main types: neutrophils and monocytes that I've already discussed in the part 1 of my presentation. Eosinophils, they have a large granules which release poison to kill pathogens, and they are important cells in allergic reactions and asthma. Then we have basophils and lymphocytes. So basophils are produced in the bone marrow but are found in many tissues throughout the body. They release granules of enzymes to destroy pathogens, and they also prevent blood clotting. They contain heparin, which is naturally occurring blood thinning substance in the body. Basophils are also involved in allergic reactions when they release histamines. Lymphocytes, these are a type of immune cell that is made in the bone marrow and is found in the blood, spleen, tonsils and the lymph tissues. The 2 main types of lymphocytes are B lymphocytes, B cells, which produce antibodies, and T lymphocytes, known as T cells. So let's take a look at T cells. T cells have specific receptors on their surface. They are activated by antigen-presenting cells after infection, and then they release substances into the blood which can signal for B cells, macrophages and eosinophils to grow and help the immune system attack. Activate phagocyte cells to destroy cells by phagocytosis, produce cytotoxic T cells during a viral infection. There are 3 types of T cell lymphocytes. You have cytotoxic T cells, which bind together to the infected cell and release a substance called perforin, which make holes in the cell. An enzyme that can be produced which moves through the hole into the cell then destroys it. Suppressor T cells, they suppress both helper and cytotoxic T cells in order to stop them from causing too much damage to the body's own tissues. Helper T cells activate most of all the other immune system cells. When it detects a virus, for example, it divides and reproduces to produce memory cells, and also activates cytotoxic T cells, macrophages, and activates B cells to produce antibodies to fight infection. Helper T cells have a protein called CD4 on their surface, which plays an important role by binding the molecules which help the immune system recognise foreign substances within the body. So let's take a look at B lymphocytes. They are also produced in the bone marrow. When B cells attach to an antigen and process it, they produce membrane markers that a T cell can then understand. B lymphocytes present these substances to T cell receptors, causing T cell activation. Each B cell has a receptor that will connect to only one antigen shape. An important difference between the cells is that B cells can connect to antigens right on the surface of the invading virus or bacteria, unlike T cells, which can only connect to the virus antigens on the outside of the infected cells. When it connects to its specific antigen, a helper T cell releases chemicals that stimulates the B cell to divide many times, with the perfectly shaped B cell receptor to connect to the antigens on the surface of the pathogen. Many of these B cells are then quickly turned into plasma cells, which are what actually make and release antibodies that connect to the same antigen as the original B cell receptor. Plasma cells make thousands of antibodies per second, which can spread around the body quickly to attack pathogens. Antibodies bind to the antigen and can destroy the pathogen directly or by preventing them from binding together or entering the body's cells. As you can see on the diagram on the screen, they bind to the antigens, coat the pathogen, stimulating the removal of pathogen by macrophages and other cells. When they are not fighting an infection, these cells move to the lymph nodes where they remain until they are then activated when the type of pathogen with the specific antigen enters the immune system again. These are known as memory cells. So let's take a look at immunoglobulins, or antibodies, are glycoprotein molecules which make up an important part of the immune system. There are 5 classes of immunoglobulins. IgA plays an important role in the cellular defense of the soft, moist mucosal surfaces of the body, such as inside the mouth. A lack of IgA often causes major infections of mucosal surfaces such as the mouth, throat and the lungs. IgD is an uncommon molecule whose true function in the immune system still remains unknown. IgE is the level of IgE in the body increases significantly as a response to parasitic infections and allergic reactions such as hay fever. IgG is the most abundant immunoglobulin and is one of the major activators. IgG molecule is able to stimulate a very direct attack on anything that it recognises as being foreign. The only type of immunoglobulin that is transferred across the placenta from the mother to the fetus, providing unborn children with some protection from diseases to which they have not yet been exposed to. IgM is the first immunoglobulin synthesized in an antibody response. IgA, IgG and IgM are found in large amounts in the human body, but they all contribute to the human immune system response. They are Y-shaped, as you can see from the illustration below. So let's take a look at self-recognition and tolerance to our own tissues. So to be able to recognise self and non-self tolerance, the immune system uses the antigens. The antigens allow the immune system to distinguish between self and non-self, ensuring that only non-self are then to be attacked. Cells within the human body show specific types of antigen that the immune system recognises in being self. The immune system will encounter the antigen of a red blood cell on the surface. Therefore, it will not be attacked because the antigen is recognised as being self. If the antigen has not been recognised before, it's then classed as non-self. The adaptive immune system response will follow by B and T cells, as I discussed before. Now, there's not a consistent level of response in the immune system, therefore the response is generated by the level of similarity between the antigen and the self-antigens within the body. So, failure of self-tolerance. So, hypersensitivity reactions occur when the normally protective immune system responds abnormally, potentially harming the body. These are classified into 4 different types. For example, type 1 are immediate reactions such as allergies and asthma. Type 2 referred to as cytotoxic, as they involve antibodies that are specific to particular tissues within the body and cause destruction of cells in these tissues, for example, autoimmune hemolytic anemia. Type 3, the binding of antibodies to circulating organs leading to serum sickness. And type 4 are hyposensitivity reactions such as multiple sclerosis and rheumatoid arthritis. So let's take a look at the rejection of transplanted organs within the body. So transplantation is the process in which cells, tissues or organs are transferred typically from one person to another. This would be carried to replace a damaged organ with a healthy new organ from a potential donor. In some cases, the immune system can potentially see the new organ as a foreign object within the body because the antigens on the cells of the organ may be different, therefore they are recognised as being non-self. Rejection can be formed in 3 different types. We have a hyperacute rejection, which will occur in minutes after the transplant. It's extremely crucial and the tissue must be removed instantly to prevent death. This type of rejection is a result of a person being maybe given the wrong blood. Acute rejection can occur between 1 week after transplantation up to a month. All people who received a transplant may occur some acute rejection at some point, and chronic rejection has the ability to take place over many years. This response is the immune system response against the new organ in the body, therefore it can slowly damage the transplanted tissues or organ. So then we have immune system disorders within the body. So immune system disorders cause abnormally low activity or overactivity within the immune system. So I'm now going to give 2 examples of disorders that can be within the immune system. So first of all, as you can see, we have rheumatoid arthritis. Now this is a long-term condition causing pain, swelling, stiffness within the joints. There may be periods which symptoms are worse, which is also known as flare-ups. Symptoms such as pain, stiffness, swelling, redness, high temperature, and possible risk factors that may lead to this immune system disorder are your genes, so possibly inheritance running within the family, hormones within the body, and possibly smoking, as there has been some evidence that suggests that people who do smoke have a higher risk of developing rheumatoid arthritis. Blood tests can diagnose rheumatoid arthritis alongside scans and assessments of physical ability. Then we also have multiple sclerosis. So this is a condition that affects the brain and spinal cord, and a wide range of potential symptoms include problems with your vision, arm or leg movement, sensation or balance. It is a lifelong condition that can cause serious disability, and people who suffer do tend to have a reduced life expectancy. So multiple sclerosis, also known as MS, is one of the most common causes of disability in younger adults. Symptoms can include fatigue, vision problems, tingling and numbness, muscle spasms, mobility problems, pain, depression and anxiety, sexual problems, bladder issues and potential bowel problems, and speech difficulties. Now, possible risk factors include, again, your genes, so running through the family, lack of vitamin D, so sunlight, again, smoking, teenage obesity is also a cause factor, and viral infections, and it is more common in women. Diagnosis for multiple sclerosis can include MRI scans, neurological examinations, blood tests, and lumbar puncture. So let's take a look at vaccination. So a vaccine is a biological preparation that provides active acquired immunity to a particular infectious disease. Acquired immunity is immunity you develop over your lifetime. It can come in from a vaccine, exposure to an infection or disease, or another person's antibodies. Acquired immunity can be split into 2 types. You have active and passive. So active immunity is the immune response to a pathogen. It relies on the body making antibodies, which take time to mount an attack against bacteria or viruses. This type of immunity lasts longer than passive immunity, and there are genuinely a few side effects. There are 2 types of active immunity. You have active natural. Antibodies are produced after being exposed to a pathogen, as discussed earlier in the presentation. When T cells and B cells are active and result in an immune response where pathogens are destroyed or neutralised by the body. You have active artificial, which are antibodies made up from getting a vaccination, which will be discussed later on in the presentation. And passive immunity occurs when antibodies are introduced to the body, such as breast milk. This type of immunity produces an immediate, though short-lasting response, as the body does not produce memory cells that are reactivated if the pathogen invades the body again. There are two types of passive immunity. So natural, antibodies transmitted from one person to another, such as breast milk via the placenta. And passive artificial, where antibodies are given to a person in the form of treatment by an injection or transfusion of antibodies. These immunoglobulins treatments are antibodies that are usually used to treat people at risk of infections, like a baby born to a mother with a disease such as hepatitis B. These antibodies are made in a lab or come from other people or animals. There are often more side effects with this type of immunity. So, immune response to vaccinations. Vaccines play an important role in keeping us healthy. They protect us from serious and sometimes deadly diseases and immune responses that can be very quick. They are based on the idea of immunological memory, the ability of the immune system to respond to a pathogen that has been previously encountered in a quicker and more effective way than during the first encounter. So vaccines work by stimulating our immune system to produce antibodies without us actually becoming infected with the disease. This is called the primary response. It is a fairly slow response as the body takes time to react to the pathogen and produce antibodies to it. If the vaccinated person then comes into contact with the disease itself, their immune system will recognise it immediately and quickly produce the correct antibodies needed to fight it. This is called the secondary response. There are different types of vaccines. They can be made from live pathogens that are treated to make them harmless, such as the MMR vaccine. Harmless fragments of a pathogen, for example, messenger RNA vaccines, such as one of the COVID-19 vaccines. Toxins, poisons produced by pathogens, such as the tetanus toxoid vaccine. Deadly pathogens, such as hepatitis vaccine and the influenza vaccine. Boosters may be needed against these as their effect can be fairly short-lasting. All of the vaccines are harmless, so a person would not develop the disease after being vaccinated, and all cause an immune response, so antibodies are produced against the pathogen along with memory cells. So after having a vaccine, if the pathogen invades the body, antibodies against it can be produced very quickly. So let's take a look at the benefits of a vaccination. So vaccines have been an invaluable tool in reducing the rate of disease and death. Childhood deaths have decreased as children are now vaccinated against many diseases that they used to die from, such as measles and whooping cough, and adult deaths have also been reduced, for example, the recent events with COVID-19 vaccine. But I'm going to focus on measles vaccine to show some of the benefits of the vaccination. So measles is a highly infectious viral disease which can lead to serious complications. Vaccines have reduced the number of deaths from measles. Since the single measles vaccine was introduced in the UK in 1968, Public Health England estimates that 20 million measles cases and 4,500 deaths have been avoided now in the UK. In the period from 1940 to 1968, where the single measles vaccine was introduced, there were 11 million notifications of the measles infection. At a rate of 300,000 per year over the 29-year period, there were 7,000 deaths in the period, with 5,000 occurring in the 40s, 1,500 in the 50s, and 767 from 1960 to 1968. Following the introduction of the single measles vaccine and leading up to the introduction of the MMR, which is the measles, mumps and rubella vaccine in 1988, there were 2 million notifications, a rate of 100,000 per year over the 20-year period. Now, deaths fell from 85% to about 20 per year. However, between 2001 and 2013, there was a sharp rise in the number of UK measles cases, and 3 people died. Number of cases have fallen since 2013, but rates of measles are still higher than they were in the late 90s. The majority of measles cases have been in people who are not vaccinated, especially young people aged 15 and over who missed out on the MMR vaccination. This was due to the inaccurate MMR controversy when the MMR vaccine was incorrectly linked to autism. Some parents chose not to vaccinate their children, and due to this, by 2003, the MMR coverage was down to 82%, lower than what is needed for herd immunity. So let's look at herd immunity. This occurs when a high percentage of the population is vaccinated, making it difficult for infectious diseases to be spread. So because there are not many people being infected, it's highly unlikely that you'll meet someone with the disease, so therefore we just say the disease is contained. In some cases, such as smallpox, this can result in a disease being completely eradicated within the population. Some pathogens are more infectious than others, so measles requires 94% of the population to be vaccinated, whereas rubella and diphtheria only require about 85% of the population to get herd immunity. However, the immunisation rates fall, herd immunity can break down, leading to an increase in the number of new cases, as we saw in the UK with measles in the early 2000s. Other problems facing providing herd immunity can be because there are other different varieties of the pathogen, or the new pathogen may mutate, often such as influenza virus. New vaccines are needed yearly to fight these pathogens because of the religious or ethical reasons of because concerns or other side effects. In summary, vaccines are an essential and effective part of the controlling the spread of pathogens, and their effects can be seen by analysing data. Vaccine programmes have improved massively around the world.